I am Harsh Doshi, and I'm going to be talking about my master's thesis project, which I did under the supervision of Professor Apratim Chatterjee at IISER Pune. The thesis is titled Segregation Kinetics of Topologically Modified DNA Polymers in Confinement. I'll explain what all of these terms mean during the presentation. Let us start with what polymers are. So this is the kind of image that pops into our heads when we think of polymers. It's a long chain of repeating chemical monomeric units that are bonded together. From a physics perspective, we think of polymers in a similar way, but we ignore the chemical structure of each monomer and instead model them as a nondescript sphere or blob. This sort of framework is more general and can be applied to many different types of polymers. Because polymers are so long, they have a large number of degrees of freedom, and thus they show interesting properties, one of which is what this thesis is based on. In order to understand what this property is, let's consider first a very simple case of just two gases that are taken together in confinement. So they're placed in a cylindrical box initially such that the two gases are on two different halves of the box. If I let this system evolve naturally in time, we can expect what will happen. The two gases spontaneously mix together. The reason for this is because each of the gases wants to maximize its entropy by occupying the entire volume of the cylindrical box. However, if I do the same thing for polymers instead, so here I have taken two polymers that are initially again placed in the same cylindrical box and are initially mixed together such that they overlap along the long axis of the cylinder. And now, if I let the system evolve naturally with time, we'll see eventually that the two polymers segregate. That means that the two polymers have moved to two different halves of the cylinder. This is in complete contrast to the gas case. But the reason why this segregation happens is actually the same reason that the gases mix. It's because entropy is increasing in this process. To understand how this occurs, let's consider a two-dimensional scenario of the same two polymers that are placed in a cylinder. However, the cylinder is now infinite in length. I've shown two cases here, one in which the polymers are overlapped or mixed along the long axis, and one in which they're segregated. In each of these cases, I can explicitly list and count the total number of conformations that the polymers can take. And if I compare the number, I'll find that the number of conformations is much higher in the segregated case as compared to the mixed state. An intuitive way to see this is that in the mixed state, the two polymers sort of restrict each other's conformations by not letting them explore conformations that can span the entire width of the cylinder, for example. In the case of the segregated polymers, the two polymers are separate. So they are free to take conformations irrespective of the other polymer. And thus they show a higher number of conformations. As a result, the conformational entropy is higher in the segregated state. And consequently, the free energy is higher in the segregated state as compared to the mixed state. Thus, going from the mixed state to the segregated state involves a decrease in free energy, which makes this process spontaneous. The same argument can be extended to three-dimensional case as well, and also for a case in which the length of the cylinder is finite. As long as the length is not too short. If it is too short, like here, then again, even in the segregated state, the two polymers would restrict each other's conformations in this way, and there would not be any entropic benefit for the polymers to remain segregated. Thus, they would remain in the mixed state. This sort of polymer physics entropy-driven mechanism has also been used to explain a phenomenon in biology. Here I've shown an illustration of a E. coli bacterial cell, and this yellow stringy substance is the DNA or chromosome content of that cell. During the life cycle of such a cell, it must grow and divide. And for that, first it needs to duplicate its chromosome content into two copies like this. And then it needs to segregate into two different halves such that the cell can divide in the middle. How this segregation occurs had puzzled scientists for some time because they could not find any explicit protein or cellular machinery that can actually pull apart these chromosomes to two different halves of the cell. Later, it was postulated that the entropic polymer physics mechanism that drives segregation of polymers in confinement is actually what aids the chromosomes themselves to segregate because they are essentially long polymers present in the cell. Whatever I've told so far has been quite well established in the field, and what I'm looking at in this thesis is to study the effect of additional topological effects of the polymers on this segregation. So people have found that there are chromosomal loops that are formed on top of this DNA contour by special proteins. How can such loops affect the segregation rate is what this thesis is trying to answer. And in particular, can such loops actually help in faster segregation of these polymers in confinement? So in order to answer this question, we form simulations of these polymers where our polymer is modeled as a bead spring polymer. Each of the monomers is taken as a spherical bead with short range repulsion between them such that they cannot overlap at the same place. And consecutive connected beads are connected via springs such that they remain connected and the polymer remains connected along the contour. This polymer is evolved in time using the Langevin equation via a process called Langevin dynamics. Here I'll explain what sort of topologies or types of polymers that we use in our study. The simplest one is the ring polymer, and it can be obtained by joining the two ends of a linear polymer. We can impose additional complications by introducing a crosslink between specific monomers as shown here. So this will alter the shape or topology of the polymer in such a way such that there are two smaller subloops present in the polymer now. We can keep adding more number of crosslinks to impose more number of these smaller subloops, as shown here. Remember that we want to study the effect of such smaller loops on the segregation of these polymers when placed in confinement. So we take two such polymers of identical topology and place them in a cylindrical cell like this, initially such that they're mixed. Then we let the system naturally evolve with time via the Langevin equation and wait till the system achieves segregation. We measure how long it takes for it to go from this mixed state to the segregated state. And we actually perform these simulations in 50 independent runs, just so we can get good statistics to compare these segregation times across various architectures or topologies. I mentioned that we need to initialize the polymers in a mixed state in a confined space, but this has proven to be quite a challenging task during this thesis. For one, because we are considering closed polymers like these, there is a possibility that if you're not careful enough, the polymers can get concatenated like this, and that could affect their segregation rates. So in order to avoid these concatenations, we start with the polymers in a big cylinder such that they're open in this open conformation where we can explicitly say that the polymers are non-concatenated. Then we gradually shrink the cylinder such that the polymers remain non-concatenated till the desired confinement is reached. However, in confinement, the polymers prefer to be in a segregated state. So if we do not do anything else, they will naturally occupy this segregated state and we would not obtain a mixed state from which we can start segregation. In order to avoid the polymers already becoming segregated while the shrinking is happening, we impose additional constraints. For example, one such constraint is that we fix the centers of mass of each polymer at the center of the cylinder, such that they cannot move apart to achieve segregation. In the presence of such constraints, even during the shrinking, when the final confinement is reached, the polymers still remain overlapped along the long axis, as shown here. And so we obtain a non-equilibrium, non-concatenated mixed state from which we can start segregation. Now, because we introduce these complicated constraints and the shrinking procedure, there can be artifacts that have crept into our system. Just to ensure that nothing abnormal is happening in our polymer system, we have calculated and plotted various monomer density quantities, which indicate that the densities are as per expectations. Moving on to the segregation, again, we want to start from a mixed state, which is obtained by imposing the additional constraints. And once we remove that constraints, the system is free to evolve to the segregated state. And we measure how long it takes for it to go from the mixed state to the segregated state. To exactly quantify when the polymers have achieved segregation, we track the centers of mass of each polymer. So here in this graph, I have shown on the y-axis the z-coordinate of the centers of mass of each polymer as a function of time. Initially, they're close to each other at 0, which corresponds to the mixed state. And then after I release the constraints, they move such that till they reach some maximum separation, which corresponds to the segregated state. The same information is shown in this graph, just that I've taken the absolute difference between these two coordinates. So again, it starts at 0, which corresponds to the mixed state, and achieves some maximum separation eventually. When it first achieves this maximum separation and stays there for some time, is when I call my system segregated. And this red line is what is indicated as the time of segregation. Remember, we get 50 independent segregation times from the 50 independent runs for each topology, which we then use to compare across various topologies. So here is one such comparison. I consider a topology of this form with one big loop and one smaller loop, and I vary the size or the ratio of sizes of these two loops to affect how that affects the segregation. On the y-axis here, I've shown the segregation time, and on the x-axis is the various topologies by varying the size of the smaller loop. Each of these boxes corresponds to the spread of segregation time obtained for that particular topology. So 0 here just indicates the simple ring polymer, and 10, 20, 60, 100 indicates the number of monomers present when an additional smaller loop is placed. In each of these cases, we can see that there's not much difference in the spread of the segregation time. A more interesting case is when we have multiple of these smaller subloops. So here, I've shown the same sort of comparison. However, I have compared now topologies with varying number of the smaller subloops. 0 just means the simple ring polymer. 1 is something like this. 2 is something as shown here, and so on. Here we can clearly see that as we increase the number of smaller subloops beyond 2, the segregation time decreases significantly. And the reason for this is actually the same reason that the polymers do not want to stay mixed entropically. Because each of these polymers repel each other entropically, even the smaller loops in the same manner do not want to stay mixed with other smaller loops due to entropic reasons. So when there are more number of these smaller loops present in the system, there are more of these repelling elements, which tends to increase the rate of segregation and resulting in a decrease in segregation time. We also perform this sort of analysis for the different initialization conditions that I briefly talked about earlier. And most of them follow the same trend. On increasing the number of smaller subloops, the segregation time decreases. This held even when we tried with systems of larger polymers with 500 monomers in each polymer. There were certain cases where this trend did not show, and we then investigated and found that the initialization itself was biasing our mixed state, which led to abnormal segregation times for particular architectures. However, in summary, what we have shown is that the introduction of topological modifications in the form of loops on polymers can help speed up their segregation when they're placed in cylindrical confinement. This sort of topological modifications that is seen in bacterial cells can perhaps help explain the timely segregation or the efficient segregation that they perform during their cell cycle. And this sort of polymer loops, if they're achieved in practice in synthetic polymers, can also have applications when such synthetic polymers are placed under confinement, artificial confinement. Thank you.